In this video, we'll go over the answers to the problem sheet from March 26. And the chapter we're looking at is kind of bringing together all of the metabolism we have learned about this semester. And now we're seeing how different organs, different signals all regulate these processes. So we're kind of zooming out from the biochemistry and looking at where these processes are happening inside the body. So first, we're going to start with just a general question. Anabolic and catabolic pathways for a given biomolecule almost never involve identical sequence of reactions because, and we talked about this before, if you had the same sequences of reactions, it would just be a futile cycle. You would go down one pathway and then go right back up. So, for example, glycolysis and gluconeogenesis. That's why we have three enzymes, 1, 3, and 10, that are far from equilibrium, and they're different in glycolysis and gluconeogenesis. We can regulate each pathway independently of each other, and so we just don't have a futile cycle breaking down glucose, building glucose back up for a net loss of 4 ATP. All right, so here is our big map of our cycles of metabolism. You can see right here, acetyl-CoA is a comic intermediate. What can we use acetyl-CoA for? So let's take a look at proteins. Can we go proteins to lipids? So proteins broken down to amino acids acetyl-CoA, acetyl-CoA to fatty acids, and lipids. Yep, we can do that. How about sugars to triacylglycerols? So glycogen, G6P, pyruvate, acetyl-CoA, fatty acids, triacylglycerols. Yep, we can do that. How about lipids to glycogen? Triacylglycerols, fatty acids, acetyl-CoA. And now we can't get to glycogen easily because remember, pyruvate to acetyl-CoA using the PDH complex is irreversible, so we can't go back up that way. Now, it is possible to go a very convoluted method. Acetyl-CoA goes through the citric acid cycle, becomes oxaloacetate. Oxaloacetate is taken out of the citric acid cycle, made into pyruvate. Pyruvate then goes to glycogen. So, possible, but we're looking for more direct. So we're going to say no, that's not really possible for us. So we're going to go with A and B. When doing this, if you said A, B, and C, I would say that that's definitely reasonable by following this pathway. But we're looking more for like directly, like acetyl-CoA directly goes to lipids. Acetyl-CoA can directly go to proteins. Acetyl-CoA doesn't really directly go to glycogen. It has to go through the citric acid cycle. Right? So, bunch of true and false. Insulin increases the intracellular concentration of glucose in hepatocytes or liver cells, and we'll see later on in these slides that that's true. Glucagon, which means glucose is gone, is secreted in response to low glucose blood levels. Yeah, that's in the name. Insulin increases the capacity of the liver to synthesize glycogen. Yeah, so insulin means you are well fed, so liver will start storing glucose as glycogen. Insulin has the opposite effect of epinephrine and glucagon. Yes, so epinephrine's fight or flight, glucagon means glucose is gone, insulin means you're well fed. Epinephrine and glucagon will try to make ATP. Insulin will say, well, let's start building stuff, start storing that ATP. Glucagon increases triacylglycerol synthesis. False. Glucagon means you don't have glucose, you're running low on energy, so it would be pointless to make fats at that point. Right, what does brain use for fuel? They mainly use glucose, ketone bodies if you don't have glucose available. And a lot of that energy goes to just a plasma sodium potassium ATPase pump. because the brain is sending out signals, so it makes sense that most of your energy is going to membrane potentials that allows you to do these nerve transmissions. What is the muscle's major energy source? Glucose. It can also use fatty acids and ketone bodies, and it can use its stored glucose, which is in the form of glycogen. What's that mainly used for? Well, muscles do contraction, so it's mainly used for contraction. How can muscles regain their ATP? Phosphocreatine is a high energy phosphate compound that can be used to remake ATP. What's the main function of the adipose tissue? Well, they store and release fatty acids, and they will also secrete hormones that are involved in regulating metabolism. And that's really due to the amount of glucose in your body, like how much fat it will release and how much it will store. What's the main function of the liver? Liver is a main player in this. As you can see, it's in the center of everything, and all the arrows that we have going here are more or less going through the liver. Like the one exception is glucose from the kidney to the brain. Right, so the liver makes sure that you have the fuel, the proper levels of fuel, especially for the brain. It is the brain's blood buffer, so make sure you have all the fuel and also take in nutrients other than fats. Why is it important that liver has glucokinase? Or I guess the question, why is it important that glucokinase has a different KM than hexokinase? Another way I could ask is, why is it important that liver has glucokinase rather than hexokinase? First, let's define what these two enzymes do. They do the first step in glycolysis, glucose to glucose 6-phosphate. Hexokinase works at a higher rate with lower glucose than glucokinase does. That's in our KM, if you remember from our kinetics back in Biochem 1. KM is 5 for glucokinase and probably like 0.1, if not lower, for hexokinase. So remember in the previous slide we just said liver stores glucose. That's one of the things it does, and to regulate the amount of glucose. So if liver had hexokinase, it would compete for blood glucose with all the other tissues in the body, which wouldn't make sense because liver is supposed to be regulating this, and liver doesn't really even use glucose all that much for energy. So rather, we have a different form of hexokinase called glucokinase, which when we're in the well-fed state, glucokinase is becoming more and more active. The more glucose we have, the higher, faster this is, the closer to Vmax it is. And so the liver will start taking glucose out of the bloodstream and start forming glycogen, which is good because we want to store it. So that's why it's important that we have glucokinase in the liver instead of hexokinase. What's the main function of the kidneys? Kidneys is main function is to filter out urea, but it also will recover important metabolites like glucose and can send that glucose to the brain. It will also excrete hydrogen and ammonia. How does the Cori cycle work? So a lot of words here, but it's super simple. Muscle breaks down glucose or glycogen to lactate. Remember we talked about this. Muscle is working, and so anaerobic glycolysis is way faster than aerobic, so muscles are just going to go to lactate. We have all this lactate build up though, so we need to take care of it. So we're going to send that through the bloodstream to the liver. And the liver is going to go through gluconeogenesis, turn that back into glucose, and send it back to the muscle to use again. So it's a nice little cycle of recovering our lactate to remake it into glucose and giving it back to the tissues that need it, the muscle tissues, which are working hard. How does the glucose-alanine cycle work? Basically the same thing, but a little bit different. Glucose glycogen is broken down to pyruvate, and sometimes during transamination, which we talked about in the last set of videos on Tuesday, transamination will happen where the NH3 group will be given to pyruvate instead of alpha-ketoglutarate like we talked about. This changes pyruvate into alanine. Well, this alanine can be sent to the liver where the ammonia is taken off and made into urea, and we'll be left with pyruvate. We're just doing the opposite reaction. Well, we want to use this pyruvate. Pyruvate has a lot of carbons, a lot of energy left, so we can go through gluconeogenesis, make this into glucose, send it back to the muscle or wherever it needs to go. But the glucose-alanine cycle is sending it back to the muscle, so it can be used for energy again. I forgot to put my question here, but the question was, how are pancreatic B cells, how do they release insulin? And that's because these pancreatic beta cells have glucokinase as well, so they only work at high levels of blood glucose like we just talked about. When they go through glycolysis and oxidative phosphorylation, almost all of the ATP is used to induce a potassium channel. So this potassium channel will start to pump, or will close rather, and the charge of the membrane will change, and that will cause it to release calcium, just like a nerve impulse that we talked about in Biochem 1. So the calcium channel will open, and this release of calcium will have a chain reaction in which insulin vesicles that were in these beta cells will go to the membrane and be released into the blood. And now you have insulin in your blood telling the rest of your cells, hey, the pancreas has realized that our blood is full of glucose. We are well fed. Start taking that glucose up. Start building your storages of energy now. Now's the time to do it because the pancreas said so. So that's how insulin is released in when you have high blood glucose. Right. What is GLUT4 and how is it activated? GLUT4 is a glucose transporter. It will take glucose from the bloodstream and put it into your cell. And it's only activated only on the plasma membrane when there's insulin around. So it's an insulin-dependent glucose transporter. What effect does insulin have on muscle, adipose tissue, and the liver? So we have 3 questions. It's basically just telling you, hey, you should probably know this table. If you don't know this table, it might be a bad time for a couple of questions when it comes to the test. So let's look at insulin. Glucose uptake, glycogen synthesis in the muscle. Makes sense. You are well fed if insulin's around. So muscle will start taking that and storing it when you need to do some exercise. Fat tissue. Start uptaking that glucose and turning it into fat, lipogenesis. Don't break down your lipids. We have plenty of energy, so decrease the amount of lipolysis. Liver, it's kind of the same idea. Start making glycogen. Start making lipids. Don't make new glucose, so decrease the amount of glucose. But glucagon. Glucagon has no direct effect on the muscle, but as we talked about in the lecture video, it does have an indirect effect. Adipose tissue, if glucose is gone, glucagon, start breaking down your fat and releasing that into the bloodstream. Liver, if you receive glucagon, that means glucose is gone. Stop making the storage glycogen. Instead, increase breaking down glycogen or glycogen lysis so we can release that glucose into the bloodstream. And there was one more. I believe I should have had epinephrine here, and it's the same idea. Epinephrine, fight or flight. Increase in the muscle breaking down glycogen, get that glucose ready for anaerobic breakdown. Adipose tissue, start breaking down the fat. We are going to either be fighting or flighting, so get that fat ready for ATP production. Start breaking off fatty acid tails. Liver, kind of the same idea as glucagon. Stop making glycogen. Start breaking down glycogen lysis. Start making glucose because your muscle's going to need it because we're going to do something here quick. So start making glucose so we can send it out into the bloodstream. All right, signal transduction pathways, what are the advantages? Like I said, we are not going to memorize all the proteins involved in this. I'm not going to torture you for that. But generally, why are signal transduction pathways seen in biochem, molecular cell bio? So this allows external stimuli or multiple signals with different magnitudes, or allows your cells to respond to these with different magnitudes and durations, because you're turning on a bunch of different proteins, and this will go and turn on other different proteins as well, and depending on the signal, it will amplify that response more and more and more. You keep getting the signal, that response will continue to be amplified. So it's a handy way for like one signal or a combination of signals to have a big effect inside the cell, because it's almost like dominoes. You knock one over, a bunch of other are going to fall in response to that. That's kind of like how a signal transduction pathway works. How are AMPKs activated, or AMP-dependent protein kinases? Well, they have a gamma subunit that will be activated by AMP, and I didn't ask this, but how are they inhibited? ATP. So AMP-dependent protein kinase, well, activated by AMP, low energy. What effects do AMP have on these next two slides, basically? Cardiac muscle, liver, skeletal muscle, and adipocytes. So in your cardiac muscle, they'll activate PFK2, which will increase the amount of F26P, which will turn on PFK1 and ultimately glycolysis. So know that it turns on glycolysis via PFK2. How about liver? Turns off fatty acid synthesis by inhibiting ACC. Turns off cholesterol biosynthesis. Don't worry about remembering hydroxymethylglutarol-CoA reductase. We didn't really talk about that. It will also inhibit glycogen synthase, which is, it will stop making glycogen. How about in your skeleton muscle? Again, inhibit ACC. So inhibit your fatty acid biosynthesis, increase GLUT4. Your adipocytes, you will inhibit lipase, triacylglycerol lipase, which again, like I said in the video, this seems odd. We think, at least I never studied this, but people smarter than me think it's because you prevent toxic buildup of fatty acids. I would never guess this, but that's what we see, and science isn't always about what you think, it's about what the results think. So it will stop hormone-sensitive triacylglycerol lipases. Leptin, funny little molecule, leptin. What is leptin? What does it do? Leptin's your full hormone, more or less. If leptin's around, that means you're full. If you don't have leptin around, you don't know if you're full, and you will just overeat. So after a meal, when you're feeling full, that's because you have released leptin. You can become leptin insensitive, and that's one reason why people think, or scientists think, people become obese. They make way too much leptin because they have way too much fat, and our body just stops responding to it. Alright, so that's your audio/video answer key. Again, feel free to reach out if anything is still confusing you. I will be more than happy to take as much time as you need in a video conference or via email to answer any questions. Otherwise, I hope to see you sometime, and hopefully I'll see you again in the next video.